and welcome to Pharma Television News Review here at BioPharm America in Boston. On this show, I have Stephen Russell and Ed Damiano. Welcome to the show.
Thank you.
Ed, I'm going to start with you. You're a professor of biomedical engineering at Boston University. We're going to talk about an artificial pancreas, in fact, an artificial endocrine pancreas. And it was your early work that led into this research we're going to talk about. Could you give us the background to that?
Sure. When my son was 11 months old, he was diagnosed with type 1 diabetes. And it was at that point that I realized that I potentially had a role to play in improving the lives of people with type 1 diabetes. I was motivated to find a way to improve the therapy in which people are managed with diabetes. So I started working with a graduate student of mine, Firas El-Khatib, to develop a technology which automatically infuses insulin, which lowers blood glucose, to automatically regulate my son's blood sugar, anybody with type 1 diabetes. So we wanted to develop a general algorithm, mathematical formulation that makes therapeutic decisions every 5 minutes or so to automatically infuse insulin, thereby removing the clinician or the caregiver from the loop. So essentially you have to sense blood sugar and get blood glucose values in order to inform yourself as to how to regulate blood sugar, and you need to infuse insulin in response to that information. And there's some decisions that are being made all the time throughout the day to manage this information. And we decided this could potentially be automated. Now, there have been a lot of people thinking about this problem, so we think, several unique approaches to the problem. So we began developing this technology. And soon thereafter, we started testing it in an animal model. And when I presented those results at the Joslin Diabetes Center in the spring of 2006, I met Stephen Russell, who was in the audience at the time. And he approached me and suggested that we are ready to move toward clinical trials.
Okay. So, Stephen, you got involved in this. Tell us a little bit about your own background and why this science that was going on was of interest to you.
Well, I'm an endocrinologist that treats primarily people with diabetes. And so I have an appreciation of how difficult it is to do this balancing act that people with diabetes have to do every day. Probably not as much an appreciation as Ed and his son, but an appreciation nonetheless. It's a tremendously difficult task. They have to check their blood glucose many times a day. They have to inject insulin, usually multiple times a day. It's, despite the best efforts and the most careful attention to diet and calculating the dose, it's often not just right, because the body's not the same every day, and so they get hypoglycemic, they have to take carbohydrates. So it's like having a second job, really, for these people with diabetes. So it's a tremendous amount of work, but it's worth it because we know that if the blood glucose is kept below about 150 milligrams per deciliter on average, you can nearly eliminate the complications of diabetes. Unfortunately, it's almost impossible to maintain it at that level without relatively frequent bouts of hypoglycemia. And so that's associated with a fair amount of anxiety, especially if they occur at night when somebody's sleeping. So it's a hard job. It's needs to be done to protect their health, but it's tremendously difficult. And so the idea of having a device that never sleeps, never gets tired, can check the blood glucose instead of 6 or 8 or 10 times a day, it can check it every 5 minutes, make decisions all night long while the person's sleeping. That was incredibly appealing to me. And there were a couple points about Ed's work that really struck me, but I think the one that was really the most innovative was the use of glucagon as a counter-regulatory hormone. And that really had not been used before in this context. And if you think about it, it makes a lot of sense. This is what the pancreas does. It uses insulin to lower blood glucose. It uses glucagon to raise it. And while a lot of people know that in type 1 diabetes you're missing insulin, so you can't lower blood glucose, what's not really appreciated is that they're often, after the first couple years of the disease, they are no longer able to produce glucagon in response to hypoglycemia. So their first line of defense is missing. Sure. And this was the first system to replace both of those critical functions of the pancreas.
Right. So, Ed, you now had basically a clinical partner, somebody who could conduct some of the clinical trials, take it from out of an animal into humans. So that comes back to the further development. Did you have to now develop the technology a little bit further in order to put it into patients? For an example, going back to your mathematical algorithm, do you need to adjust that mathematical algorithm from animal to human?
Yeah, it's a good question. So we were not really sure how well the experiments that the experimental results that we obtained in the pigs were going to translate to human. We were pleasantly surprised that they translated quite well. There was one important difference that we found, and that was that the pigs that we used didn't actually have type 1 diabetes. They had a diabetes-like pathology that we induced. And one thing they didn't really have was an autoimmune disorder. So they didn't have this immune system that's that people in type 1 diabetes are suffering from this attack of the beta cells. And so essentially our pigs didn't have these antibodies to insulin, and so we suspected, what we found really was that the subjects with type 1 diabetes had much more delayed absorption of insulin than the pigs did. And we attribute it to potentially these anti-insulin antibodies that people with type 1 diabetes are likely to have at higher levels than the pigs. So that was the main difference. However, we were able to accommodate that quite easily because the general formulation of our mathematical model accounted for insulin absorption in the design of it. So the controller knows that when it doses insulin, it takes a finite amount of time to appear in the blood and then to clear. It was a very simple matter to just slow down that assumption, basically tell the controller that things take a little bit longer in people with type 1 diabetes. So we did a first round of experiments in subjects where we assumed very fast insulin absorption, like we saw in the pig. And then we came back and repeated those experiments. We had some hypoglycemia in those instances. For the people that were most delayed, we saw hypoglycemia. So then we brought all those subjects back and repeated experiments, telling the controller that everybody's absorption is slower. So we made a global change to the parameters. And in that process, we found that we could regulate blood sugar in all these subjects with no hypoglycemia. So that was the major learning point for us from the translation from animals to humans.
Right. Now, Stephen, you were conducting these clinical trials on behalf of Ed. And first of all, just in terms of this is not a simple task. Obviously, you need funding and so forth. Right. Just at this stage, what support were you getting, and where were your, how did you cost these particular trials that you needed to do?
Well, initially we had a small amount of funding from an organization called the Coulter Foundation, which has as its goal the translation of basic biomedical engineering science into the clinical realm. So that was a small amount of money that allowed Ed to do the preclinical experiments and allowed us to have the space to design a clinical trial, but it certainly wasn't the kind of money you'd need to actually conduct a clinical trial. So we went to a number of organizations, but the one that stepped to the plate to provide funding was the Juvenile Diabetes Research Foundation. And they provided funding for this, the first phase of the study, which we've now completed. And subsequently, using those results, we were able to go to other funding institutions, including the National Institutes of Health and a relatively new organization called the Helmsley Trust, to get the funding required to expand our efforts and do the current clinical trials we're conducting now.
Okay. So the clinical trials, you said have come, the first stage has come to an end. Is that correct? And what are the results from that? And what key findings came out of that?
The key findings were that with what we call a bihormonal artificial endocrine pancreas, that is one that uses both insulin and glucagon, that we could achieve really quite good blood glucose control that was safe, meaning that we didn't have any hypoglycemia in those experiments without any need to give extra carbohydrates. And what do I mean by good glycemic control? Well, we had an average blood glucose just above the 150 mg per deciliter goal that is set by the American Diabetes Association. And I should point out that that's actually better than most people with type 1 diabetes are able to achieve on their own. If you look at the whole population, the average blood glucose is substantially higher than that. It's still not quite to our goal, but the key point was that in our study, we didn't provide the controller any information about when a meal was coming. We didn't do any pre-bolusing for the meal with insulin. This was a truly closed loop. The algorithm was just reacting to what it saw in terms of blood glucose. But even in those really very, very challenging circumstances, when we provided big meals, we picked subjects that had absolutely no residual insulin secretion. We designed the trial specifically to really be a torture test for a closed loop algorithm. It still performed remarkably well. And the results of the pigs translated remarkably well into humans, except for this difference in the pharmacokinetics. And then that was easily dealt with by a tweak to the parameters. So it was tremendously encouraging, and it gave us the impetus to move on to a trial where we would get closer to the kind of device that would actually be could be approved and released into the general use.
So in the current study, we're actually made a number of changes. The most important would be perhaps the fact that the sensor that we're using is going to be a portable sensor that's more like the system you would actually envision a subject wearing. In the first trial, we actually used a glucose sensor that sampled blood from a vein, and that's not a practical implementation of this device. But there were a number of scientific reasons why it was good to make this choice and design the trial the way we did initially. But now, with the success that we had in the first trial, we move to the second trial, and we start replacing components with more realistic, practical, portable devices. And in that process, we had to make certain changes to the algorithm. But we were also testing those technologies in the pigs while we were conducting our first clinical trial. So in parallel, we were making the transition already in preclinical experiments while we were doing our first clinical trial. So that was one of the main things that came from that. So in the current study, we will be actually testing a more realistic system. I should mention, though, that also a practical way this device will likely be used is that subjects will give a little bit of insulin before their meal. And when we did the first study, we really wanted to, as Stephen mentioned, challenge the controller as much as possible and make an entirely reactive system. But we think in reality, people will use this system in a way that would favor the system a little bit more. And when they sit down for a meal, they would prime it with a little bit of insulin.
So it's a proactive, reactive device.
Proactive. And so now that's what we're testing in our current trial. So we do expect to see this average blood sugar that Stephen referred to go down, and hopefully with no additional hypoglycemia. We're also challenging the system by testing things like exercise, which also is a challenge to glycemic control.
If I can ask you, Stephen, I mean, you've worked with patients, as you said, and clearly we're talking here about modulating glucose levels, but the implications of this is not just simply convenience or inconvenience, it's a long-term outcome for the patients themselves. Absolutely. Because the ultimate side effects of having insulin are detrimental, obviously.
Well, the side effects of hypoglycemia are relatively minimal in the short run, but in the long run they can be very detrimental. The common complications, the so-called microvascular complications of diabetes, of course, are retinopathy, which can lead to blindness, nephropathy, which can lead to kidney failure and the need for dialysis, and neuropathy, which can leave people vulnerable to severe injuries to their feet that they won't even feel, and amputations. So these are all terrible complications that are very expensive to our healthcare system and very damaging to people's lives. But what we know from important trials, landmark trials like the Diabetes Control and Complication study, was that if you can maintain the average blood glucose below about 150 milligrams per deciliter, you can almost completely eliminate these microvascular complications. So that's our goal, and that's always been the goal of diabetes care. The difference is with a system like this, I think it's realistic to maintain that level of control without hypoglycemia. And that's something that's never really been practical or possible in so-called open loop control. There just isn't enough monitoring, no matter how dedicated somebody is to managing their diabetes, no matter how much time they're willing to devote to it, it's almost impossible to avoid hypoglycemia. There's a famous graph in one of the DCCT papers that shows the average blood glucose on this axis and the complications on this axis, and they go up very slowly, and then they take a sudden rise after this average blood glucose of about 150. And then companion graph is the rate of hypoglycemia, severe hypoglycemia, the kind that requires somebody else to assist you, or you could die potentially. And that graph is flat, and then it takes a sudden rise upward, and it's at about the same place. So you're walking this tremendously dangerous line between long-term complications and short-term disaster. And what I think this system could do for my patients and for Ed's son is to allow them to walk that line with very little input from themselves, and to walk that line at night when they're not paying any attention. Exactly.
Right.
For them to sleep and their parents to sleep.
Right. The controller doesn't need to.
I'm going to come back to you, Ed, about the device itself and how it needs to go forward. But just one other question for you, Stephen. This research has revealed a couple of things, I understand. One is that the current insulins could be improved upon, and also glucagon itself is not an FDA-approved drug, inverted commas.
For this application.
For this particular application, that's right.
So what do we need from the pharmaceutical industry in order to get this device up and running, really?
Absolutely. So we found a couple things. One was that the pharmacokinetics of insulin are absolutely critical. The faster the insulin is, the better control we're going to get. And current insulins are certainly faster than they used to be, and they've really provided a revolution in diabetes care. The 3 rapid-acting insulin analogs that are available today have really improved blood glucose control and reduced hypoglycemia in people with diabetes because they better match the absorption of food. However, their limitations became clear when you have a system, when you're measuring blood glucose every 5 minutes, the controller is responding, and there's this loop. You're trying to close a loop. And the more time that elapses between the time the controller does something and the effect is felt, the more conservative the controller has to be in controlling the blood glucose. So if we could shorten that time by improving the rate of insulin absorption, that would be a tremendous benefit to improving closed-loop control. The second thing is that we have found that glucagon in the very small doses that we use in this trial, and I should point out they are very small doses, as opposed to the 1 milligram dose that's the FDA-approved treatment for severe hypoglycemia, we're using 10, 20 micrograms at a time of glucagon just to slow a descent of a blood glucose as it falls toward the hypoglycemic range or flatten it out. This is not an approved indication of glucagon, and glucagon has never been approved for use in a pump like this. It's meant to be reconstituted and injected immediately. And in that current formulation that's approved, it's not very stable. It degrades over time. And so we need a stabilized formulation of glucagon that can go into a pump reservoir and stay there for 2 or 3 days, just like insulin currently does, to make this a practical system. And so not only is somebody going to have to stabilize the glucagon, which I know a number of people are working on now, but they're actually going to have to pursue this as an indication for this glucagon.
Sure.
And the potential benefit is that while glucagon is not a very it's commonly prescribed as a rescue kit, but most of those kits aren't used, and so it's not a very big market. If this closed-loop system using both were to come into general usage, glucagon would be as big a market as insulin.
So Ed, from a medical device point of view, it's very much still an assembly on the lab bench in some ways. So what are the next stages to turn this into a proper portable device?
Well, so what we've been working on is essentially moving from a system that was manually controlled and had components that weren't practical in an ambulatory closed-loop system or artificial endocrine pancreas, to something that ultimately can be miniaturized and made portable. So that involves things like changing the glucose sensor, as I mentioned before, from one that's sampling blood to one that's sampling glucose just under the skin, and also coming up with technologies by which we can infuse both insulin and glucagon. Those also have to be developed. So potentially pumps with 2 reservoirs, one that contains insulin and one that contains glucagon in a single unit, that also receives a signal from a continuous glucose monitor, all needs to be sort of built and miniaturized. Our algorithm, which currently runs on a laptop computer, not because it needs the computational overhead of such a device, but because it's convenient to work on a system like that. Ultimately, once we converge and we come up with something that we're satisfied with, that would be embedded into the logic of a pump, essentially, and that would receive input from a continuous glucose monitor. So there's the process of anticipating the kinds of devices that are going to be changed, which we've already started to do by switching the glucose sensing technology from a blood glucose to a interstitial fluid glucose device. But also we're making changes to the logic as well, to some extent, because we're now giving small boluses before meals to prime the system, which is the way we expect the system would ultimately be used. We have to think about things like what happens when the system goes offline. I mean, you have a sensor that you're wearing under the skin, and that can get pulled out. The system needs to be able to switch into a system that is similar to what people use today, an open loop system temporarily until you can bring the sensor back online. The system can't run without glucose sensing, continuous glucose sensing. And that can fail. So failure modes have to be thought about, and we've been working on those kinds of things. Challenging the system in new environments. Right now, all of our studies are in the Clinical Research Center. But even within the context of the Clinical Research Center, in hospital setting, we need to introduce exercise. The subjects have to be more ambulatory. That's the study we're working on right now. The next study we want to graduate to where subjects have free range of motion throughout the hospital. They can eat at will. They don't have to eat structured meals, but they can eat snacks. They can eat when they want. They can exercise to a greater extent or to a lesser extent. So we want that kind of variability. So we have to test it in several more inpatient studies. But then ultimately we need to move and transition to an outpatient study. And we have to design, together we'll design an experiment that will allow subjects to test this for the first time at home. And in that system, there can't be a laptop computer. It's got to be portable. It's got to be something that hangs on a belt clip, so to speak. So those are the kinds of things we're working on. We're looking at a horizon, hopefully, of getting those experiments done over the next 2 to 3 years. And then a pivotal trial would ultimately need to be done, multi-center study, in which subjects are wearing this thing for a period of 3 to 6 months. And you can really get metrics of success over a longer period of time. For that to happen, of course, the system really has to look just like the device that what we envision the subjects, people will be using. So that will involve, our partnerships with the industry collaborators that we currently have, sort of coming on board and taking more ownership of this and building the device and porting our logic onto it in sort of a permanent way. So we're headed toward that, and we hope that in about 5 years we could potentially have a commercially available closed-loop system, but that would require, several companies in the medical device industry and even in the pharma industries to step up and play a big role.
Right. So this is none of the technology that's going to be required to go into the future is really that groundbreaking. I mean, it's within the grasp of our current capabilities.
The point was to test what is the best we can do with current state-of-the-art FDA-approved glucose sensing technologies and insulin infusion technologies. That's really, we've constrained ourselves this way. We don't want to be using, technologies that are 3 or 4 steps down the road. We really want to be using current technologies, and then the closed-loop system we envision would evolve with those technologies as they evolve.
Right. So, I mean, the sensor we're using right now is an FDA-approved sensor. It's not approved for use in a closed-loop system, but it's approved for people with type 1 diabetes.
My son wears one of these.
and the pumps are also FDA approved. So, what's really new is the way we're putting them together and the algorithm that's driving them in the use of glucagon. Right now it takes 2 pumps to do that, but there's no reason why you couldn't have a single one that would do both.
But to take this forward to become a commercial product that will serve the interests of patients ultimately requires a new partnership.
Or to enrich the ones we have, and potentially new ones. So I think that it's both. I mean, we certainly have collaborations that we think will go forward, but we will indeed need some additional ones as well.
Well, there's a medical device to take the medical device forward, but then there's equally the requirements for fast-acting insulin,
right,
and a glucagon that's appropriately approved,
right,
and has the stability that's required for the device to work effectively. Right. So it's pulling those two together. And getting it, because what I've heard is a fantastic story. It's essential now that the next stage allows you to realize the great ideas that you've brought together.
We're also hoping that some of the excitement that we're generating in our preliminary results will attract the attention of the pharma industry, because that's the one group that we really haven't interfaced with too much. But I think we're laying some groundwork to show them that they have a big role they can play here. And there's a great advantage to getting involved.
And to some extent that has happened. I mean, there is interest in generating stable glucagons that I don't think would necessarily have been the case. So there's at least
news in the last few months.
2 companies that are developing stable glucagon formulations that we know of. Perhaps we also ought to put a plug in for a special issue of the Journal of Diabetes Science and Technology that we are co-editing along with Ken Ward and Jessica Castle. Yeah. And this is an issue specifically on stabilized glucagons. So
Yeah, so I mean, I think that the idea, of course, is to do these things as much in parallel as possible. So while we're working on the device side of it, we would like the pharma industries to get involved in stabilizing glucagon and pushing that through a regulatory pathway, because it's going to have its own, regulatory, issues that's going to have to sort out over the course of the next several years.
Ed and Stephen, thank you very much indeed for coming on the show.
Thank you. It's a pleasure.